Have you ever wondered what builds your muscles, enzymes, hormones, and antibodies? Proteins are responsible for these essential functions and form the molecular machines of life. Proteins are large biological molecules built from smaller units. They provide structure, catalyze reactions, transport molecules, and support cellular communication in every organism. In many cells, proteins make up more than half of the dry weight, emphasizing their abundance and central role in maintaining life and cellular integrity. To understand proteins, we begin with their building blocks, amino acids. These small molecules join in specific sequences to create long protein chains. More than 300 amino acids exist in nature, but only 20 standard amino acids are commonly assembled into proteins across all life on Earth. Over the rest of the video, we will see how these amino acids link through peptide bonds and how chains fold into functional protein structures. Each amino acid has a common backbone and a unique side chain, called an R group, which determines its chemical behavior and interactions. The backbone includes an amino group, a carboxyl group, and the central alpha carbon that links to the distinctive R group on each residue. R groups vary widely. Some are hydrophobic, others polar, and some carry positive or negative charges, directly affecting folding and function. Tiny residues like glycine provide flexibility, bulky residues introduce constraints, and charged residues form ionic interactions that stabilize structures. Color-coding residues in models. One color per property makes it easy to spot hydrophobic patches, charged regions, and potential bonding sites in a chain. Knowing individual amino acid traits prepares us to watch how they connect into chains and influence folding into higher-order structures. A peptide bond forms when the carboxyl group of one amino acid reacts with the amino group of another, creating a covalent link between residues. This condensation reaction releases a water molecule and joins the two amino acids, extending the polypeptide chain by one residue at a time. Peptide bonds are chemically planar and relatively rigid, which constrains backbone angles and helps determine the possible shapes a chain can adopt. As each amino acid is added, its side chain becomes part of the growing sequence, contributing chemical features that affect later folding decisions. In cells, ribosomes catalyze peptide bond formation, reading genetic instructions and ensuring each amino acid lands in the correct position of the sequence. Whether in ribosomes or in lab synthesis, peptide bonds create the backbone that allows amino acids to form long, information-rich polypeptide chains. Once a chain exists, it begins to fold. Folding is driven by interactions between side chains, backbone hydrogen bonds, and the surrounding environment. Primary structure refers to the exact linear amino acid sequence. It is the fundamental code that determines a protein's final shape and function. Secondary structure includes repeating motifs like alpha helices and beta sheets, stabilized mainly by hydrogen bonds along the backbone. Tertiary structure is the overall three-dimensional fold, stabilized by hydrophobic interactions, ionic bonds, hydrogen bonds, and covalent disulfide links. Quaternary structure forms when multiple folded polypeptide subunits assemble into a functional complex, often with precise subunit arrangements. Correct folding enables specific active sites and binding pockets, while misfolding can lead to loss of function or aggregation and disease. Proteins serve diverse roles: structural support like collagen, catalysts as enzymes, carriers for molecules, and regulators of cellular pathways. Antibodies are specialized proteins that recognize and neutralize foreign antigens, demonstrating how sequence and structure create biological specificity. Color-coded 3D models and stepwise animations make it simple to see amino acids, peptide bond formation, and folding patterns at a glance. Remember these essentials: 20 common amino acids, peptide bond polymerization, and hierarchical folding from primary to quaternary structure. Their diversity and abundance, often more than half of cellular dry mass, underscore protein's central role in health, physiology, and biotechnology applications. Explore further by studying sequence, structure, and function, and you'll unlock how proteins drive biology from molecules to organisms every day.